Hello and welcome to microbiology, the greatest of all the sciences. Don't tell any chemists that I said that, however, because they might disagree with me. So I wanted to start off with an overview of what microbiology is, the kinds of organisms that we will be looking at, and what the study of microbes actually looks like. Where we came from as microbiologists, a little bit of the history, and then where we're headed in terms of all this new knowledge that we have about the important role that microorganisms have in our lives, not just to cause diseases, but actually also to keep us healthy, and that of every other living thing on this planet beside them. So it's a big topic, but we have a whole course to do it. Going to start small with a public service announcement about the need for you to go back and, if you have not already done so, to read the course syllabus. The syllabus is a contract in a way. It's my contract with you. When you read the syllabus and you sign up for this course, you agreed to abide by the rules of the syllabus. So in that document, you will find all the information that you could possibly want to know about the course, in terms of how your grade will be determined, what the attendance policies are, what the course policies are, how to get in contact with me, what to expect in terms of how the semester will roll. It's vitally important for you to spend a little bit of time reading it, as our lovely meme here shows that, it's there for you to read, it's there for you to refer to, so it's vitally important that you do that. And like I said, this is a public service announcement because this is true for pretty much all courses that you will be taking in college. And, I know that college students everywhere like to refer to the first week of classes as syllabus week, because all of us professors love to just talk about the syllabus, but, there's kind of a reason why. So that's it. That's my syllabus message for you, me and the cat. Tell you, make sure you check out that syllabus. Okay, so let's move on to talk about what microbiology is. And whenever you see the word ology, immediately you go to, well, it's the study of something. And since it's microbiology, it's the study of little tiny biological organisms, otherwise known as microbes or microorganisms. So that's what it is. That's what microbiology is. And then, of course, the first question to come to mind is, all right, so what is exactly a microbe? So what do you do when you have questions like that? You Google it, and the first thing that pops up is always Wikipedia. When you look at what Wikipedia has to say about what a microbe is, it comes up as microorganism. So it is a tiny living thing, a microscopic living thing. Technically, if it's microscopic, we shouldn't be able to see them without the help of a microscope. But here's an interesting thing, a fun fact: some microbes actually can be seen because they grow in such large numbers that you can actually see macroscopic structures. So that's some of them remain microscopic. So most microbes tend to live a unicellular lifestyle, meaning that they are single cells. The whole organism, if you will, is a cell. Some are multicellular, and that's certainly true when we get into the macroscopic living things, plants and animals in that realm. So another question is, all right, so a lot of students who are headed into nursing or health sciences kind of view microbes as disease causers. They may not be present everywhere, but our job is to, destroy them, get rid of them off surfaces, and try to avoid them at all costs. The reality is we can never do that. We absolutely cannot do that, because microbes are found everywhere. And by everywhere, I mean every single location where you don't even think it would be possible for any living thing to survive. There are microorganisms, and most often those microorganisms living in those places where nothing should be living, turns out that they are in the realm of bacteria. So bacteria happen to be my favorite group of living things. Just that's a little fun fact for you to consider as we go forward. So we'll be spending a lot of time talking about the structure, the function of bacteria, and their closest relatives, which are the archaea. Okay, so, and I'm not, by the way, endorsing you using Wikipedia as a scholarly source of information, but it's really good for background searches. Okay, so moving on to, you got an idea what a microbe is. So who are the microbes? And notice that we can kind of put them into two major headings, those that are cellular and those that are acellular. And that's kind of weird because, you know, in a little while, we're going to be talking about characteristics of life. And according to every biologist ever, life is cellular. So how exactly is it that we have microscopic living things that are acellular? So that's a bit of a conundrum, and we'll deal with it when we get to that point. But right now, let's just look at, you know, microscopic living things. On the cellular side, notice that they could be both fungi, protists, you know, fungi like yeast and molds, for example, fall under the realm of microbes. Protists, protozoa, algae are considered microbes, even though they can grow to form, you know, macroscopic forms. And then there are the true microscopic living things, the bacteria and the archaea. These are the unicellular prokaryotic organisms that make up a very significant part of the tree of life. So those are the cellular types of microbes. This course focuses a lot on bacteria and archaea. And then looking at the acellular side, so the acellular microbes include viruses. They are not cellular. And that is a very interesting feature about viruses, because if you're defining life as, you know, being a cell, then where exactly do viruses fit into that? So that's a bit of a conundrum. And not only are there viruses, there are a thing called viroids. There are nucleic acids floating around out there, and there are prions, which are proteins. These are basically molecular structures made out of molecules. So, you know, it's only been in the past couple of decades that we have had the ability to study these things to any extent, and we're still trying to figure out exactly where they all fall into the grand scheme of life, based on how of course, we define it. So one thing that I want to do is just, you know, give a shout out to the microbes, because 99% of them are essential to life, at least 99%. They're found in every ecosystem. They do not cause any diseases. And in fact, they promote health and wellness in, you know, humans who care about that sort of thing, but also in the environment in general, keep things in balance. Sometimes, you know, things get out of balance, but nonetheless, they do a very good job at it until other groups of living things get in there and screw it up. So most, 99%, probably higher, are just out there making a living like everybody else. A few can cause diseases. So there are diseases that have plagued man since man has been in existence, which, you know, a couple 100,000 years since we started paying attention to diseases. There are some that rarely cause very horrific or fatal diseases, such as, for example, Ebola would fall into that category, or, you know, the plague would be one that caused, you know, very rapidly fatal diseases that everybody is terrified of. Tetanus was one that, and rabies is another one that terrifies people because of the consequences of those diseases. But that is not the majority of microbes. It's a very, very tiny fraction of microbes. And, you know, moving forward, one thing to realize is that because the horrific ones and the ones that cause sickness are the ones that impact us the most, at least at the moment, a lot of microbiology history has been based on trying to understand diseases caused by microorganisms. Okay, but there's way more to it than that, and I hope you'll be seeing that soon. So just kind of looking at this scheme and then trying to figure out, all right, so where do microbes fit overall in the grand scheme of life? And so this is something referred to as a phylogenetic tree. And it's actually more like a shrubbery as opposed to a tree. And I should say that with a British accent, because all throughout evolutionary time, all of these different groups of living things have been sharing genes back and forth with one another. So, and you'll see some overlaps elsewhere too. So that's why I like to think of it, it's not a phylogenetic tree, it's a phylogenetic shrubbery. But to keep it simple, we'll just show it as this tree where it has a root, right? There's a seed, and that seed, which is the one thing that all branches of life have in common, was a cell. So somewhere in the realm of 4 billion years ago, cells came into being. The chemistry of that is still, hotly debated, but the reality is it was probably related to physical forces in the environment and water that led to the creation of cells. And then cells figured out, hey, doing stuff is a lot easier if we do it in this contained fashion. So then about 3 billion years ago, there was a switch, not a switch, but just something happened that led to the branching off or evolution of a group called the bacteria. This is a domain, a phylogenetic domain, the bacteria, called the big B bacteria. And these are the ones, when I say the word bacteria or anybody does, these are the ones that you probably think of right off the bat. So, this is where E. coli, a bacteria that everybody more or less recognizes for various reasons, or Staph aureus, that's where these guys reside in this realm. But then notice this branch point at about 3 billion years. So these guys went that way, but then this other branch headed this way. And then there was another branch off of it. These cells, these are also bacterial cells, but they belong to a completely different domain, which we now know because we have looked at their genes and we've looked at their other properties. So they, under the microscope, they look like bacteria, but when you start looking more closely, they are quite different and distinct. And in fact, this is the branch that gave rise, if you will, to the eukaryotes. So the eukaryotes, again, would be the fungi, the protozoa, all the way up to us, plants and animals. And when you look at phylogenetic shrubberies such as this, one thing to keep in mind is that these branch points represent time. So the longer the distance between branches, the longer the evolutionary time. So you can see that, following this branch, here's our universal cell ancestor, kept going, keep going, keep going, keep going, keep going. We are about as far away in time as you can get, because we are newly invented. Humans are new inventions in this grand scheme of life, which seems kind of weird because, human history is, like I said, a couple hundred or more thousand years old. But nonetheless, we're pretty new inventions, and we obviously still have a lot of work to do. So what we're going to focus on mostly are these two domains, the bacteria and the archaea domain, their features and characteristics and why they are important to study. Again, interesting to note that eukaryotes, what the difference is between these guys and the eukaryotes. And then just please note the viruses are sitting over there by themselves because at the moment, they are genetically distinct from these. And in fact, it's a very complicated relationship that we have with viruses. And that's something that we'll talk about later in the course. But at this point, it's just safe to say there are no branches from the family tree that lead directly to viruses. So that's, like I said, it's one of those conundrums of biology. If we're going to call them acellular microbes, then how come they're not on the tree? We'll get to that. Something to look forward to. Okay, so microbes exist. Let's consider the study of these microbes and a little bit of the history. It's actually, microbiology is a fairly new science as a science, but realize that microorganisms have affected humans for as long as there have been humans. For the simple reason is that they cause the destruction of food, right? Microorganisms can destroy food supplies. They can cause illnesses that killed off whole populations. So they've been of interest to humans. And actually, you know, on the really gross side, they cause decay of humans. And that actually led to one of the theories of diseases called the miasma theory, meaning that when you die, if you were not buried, you smelled really, really bad. And that was actually because your, you know, molecules in your body were being broken down by microorganisms. But what the prevailing theory was, that those bad smells came out of your body and invaded others. So it was the smells, the gases, the chemistry that was causing all that. It wasn't, and there wasn't any, you know, any living thing involved in causing these diseases. It was this mysterious, gassy, yucky, smelly stuff. But the reality was, it was microorganisms that were causing decay. And so the, you know, the tradition of burying people to prevent those gases, the miasma, from spreading and causing more disease came into being before we even knew that there were microorganisms. So when you look at, you know, the overall history of things, humans had no idea that there were microscopic living things until Koch and Van Leeuwenhoek in the 1600s invented a tool that got called the microscope that allowed them to see beyond the scale of what our eyes could detect. And when they did that, they found out, oh, geez, you know, we're made of these tiny little things called cells. And then there's these other random cells out there. And, you know, so that was the first view of these, of microbes, as far as how microbes fit into, you know, life. Nobody really thought much about that because they were not considered alive. They were just interesting entities. And then finally, it was like, all right, they are alive, perhaps, but it took another 100 to 200 years before it became established that, you know, microorganisms had a life of their own, that they reproduced on their own. And then, of course, the connection between diseases and the presence of microorganisms. So, you know, the period of diseases, right, plagues and diseases, and just realizing that diseases could be caused by microorganisms and coming up with scientific ways to study them actually began with Louis Pasteur in the mid 1800s. And from that point forward, there was this, the so-called golden age of microbiology, which led to major breakthroughs in not just the field of microbiology, but also medicine. So evidence-based medicine has its roots in this golden period of time for microbiologists. And, you know, it allegedly ends in the early 1900s. It really ended at the age of antibiotics because at that point, there was less interest in studying the disease process, the causes of the disease, because now we could cure them. And so we had won that war, or so we thought. Of course, now we're in the post-antibiotic era, and we're having to go right back to the beginning to look at things in different ways with regard to our relationship with microbes and their relationship with antibiotics. So, you know, during this time period, some of the good things that happened, you know, researching epidemics. So epidemics like typhoid fever and cholera killed large populations, and those populations were crowded into cities, because remember, the time frame that we are looking at here is the beginning of the Industrial Revolution. So everybody's running into the cities, living in crowded conditions, trying to make a living. And that just invites the spread of diseases. So people started trying to figure out how to stop those diseases from spreading, for obvious reasons, because they wanted to prevent them from spreading in the areas that they were out to other areas. So that's what got Pasteur started, actually. And, you know, kind of looking through this period of time, some names pop out. The two that pop out the most are Louis Pasteur and then also Robert Koch. And I would say these two gentlemen are probably considered the fathers of microbiology. So, you know, just to give you a little bit of overview about who I call these guys, the golden men, Pasteur being the most golden because he was a very interesting scientist. He was a chemist. He was actually studying fermentation, why? Because fermentation was known as a process how wine was produced from fruit juices. And so he was the one that figured out that the chemistry that turned grapes into wine was actually being done by microscopic things that were found in the mix. In the mix meaning the fruit juice. And so then he was able to show that different types of these microscopic things, depending on what you fed them. So if you fed some of them sugar, they grew and they produced alcohol. But if you fed the other guys the same sugar, they produced acids like vinegar from it. And so he was trying to figure out how to ensure that food, the sugars got turned into alcohol, because obviously the wine industry didn't want vinegar, they wanted wine for their profits. And so he figured that out. And then he also figured out that if you prevented the other guys, meaning the bacteria, from getting into the fruit juice, then you ended up with good wine every single time. And so he invented the process known as pasteurization. Big surprise there. And he did it because if you heated the grape juice up to a certain point, cooled it back down, and then you added your culture organisms, which were yeast, you ended up with alcohol every single time. If you let the other guys, the bacteria, get in there, sometimes they messed up the batches because they grew faster and they produced alcohol instead. So by pasteurizing the fruit juice, if you didn't heat it up to boiling or past boiling, then you were able to destroy the acid producing microbes and leave the fruit juice intact, so that it would, when you added the right thing to it, it would produce alcohol every single time. So you have Louis Pasteur to thank for that, if nothing else. Pasteurization was later applied to other foods like dairy, because there was a known association between the consumption of dairy products, dairy milk in particular, from farms, and certain types of diseases like brucellosis and a few others. So that got, his technology got used for that process as well. And now it's used for a lot of different foods to reduce or completely eliminate any microbes that might be found in that food. So he was one of the first to propose the germ theory of disease, meaning that diseases just didn't happen. It wasn't because somebody died and they smelled bad as they were decaying that caused the disease. It was because there were microbes that caused the illness. And he did that by not looking at human diseases, but looking at something called silkworm blight. And again, silkworms were important because they produced silk, which was very important in cloth making back in the day. And remember, we're talking about the middle to end of the 1800s. So you don't want bacteria in your fruit juice, and you don't want, fungi in your silkworms. So he first postulated that diseases could be caused by these microscopic entities by identifying the cause of silkworm blight. And then he started messing around with, all right, how do we prevent diseases? If germs are causing disease, how do we stop them from doing that? And so he was one of the first, again, to realize that if you could attenuate microbes, meaning take away their superpowers, the things that makes them evil, and inject them into animals in that state, the animal, which would apply to us too, was able to make an immune response, make antibodies against it, and that would cause protection against the infection. So he did this for the first time. So he was working in a lab and doing it in a laboratory, and he was actually approached by parents of a kid who had been bitten by a rabid dog and said, "We know you're doing science on this. Is there anything you can do for our son?" And this was in the days before there was any thought about informed consent. And the boy was going to die anyway, so he did that. He did his experiment on the young man, and he saved his life. So that was the first case of anybody ever surviving rabies, was because of Pasteur and his attenuated microbes. And once he did that, as you might expect, he became the hero of France. And his discoveries really were what paved the way to the idea that you needed to do really good science in order to make any kind of forward motion in medical practice, meaning the diagnosis of diseases, mostly treatment of diseases, because remember, in that time there were no treatments for infectious diseases other than herbs and in some cases witchcraft. So as a result of his work and as a result of donations to him, he, with others, was able to open the Institut Pasteur. And that was the place where the first course in microbiology, his microbiology, was ever taught, was at the Institut Pasteur. So that's a little bit of interesting history there. The other golden man was Koch, Robert Koch. He is a German. So he built on what Pasteur had shown with the germ theory and wanted to take it further. And so he developed an experimental model to prove scientifically that a particular microbe was a cause of disease. And here's why: because you could take a person who had a disease and try to take bacteria from them. It turns out, and he realized that, and many others too, that we are full of bacteria. So how do you know which one is the one that causes the disease? So he came up with Koch's postulates as a set of steps that you need to follow in order to prove that a microbe was a cause of a particular disease. And that also was a scientific method that made him very, very famous. In fact, he was the first microbiologist to win a Nobel Prize, one of the first of anybody to win a Nobel Prize in 1905, because he actually discovered the cause of tuberculosis and figured out how to stain it and several other things. Not him personally, but many others who worked for him in his laboratory. So everybody back in the day wanted to be a microbiologist. Americans were rushing to Europe. Wealthy Americans were rushing to Europe to get involved in this new evidence-based medicine and microbiology. So a lot of the early American scientists, medical doctors that, names you might recognize, most of them were trained in either the labs of Pasteur or Koch before they came and started to do their work in the United States. So, between those two, and I have to say something here about Koch's postulates, because to some extent, we still rely on Koch's postulates to help us identify whether a particular agent is the cause of a disease. But I want to point out that there are limitations to Koch's postulates, which got recognized pretty early, but are still under debate as to whether or not these are, the rules that we should be following in every single case. And as you can see, Koch's postulates, postulate means this is it, this is the rule. They're really more like guidelines, actually. So here are the postulates. Postulate 1, 2, 3, 4, right? So you have, in the case of a sick animal, and because he worked with anthrax as his first organism, that's what this is showing us, a sick animal, because anthrax was actually a disease of animals that sometimes cause diseases in humans. So you found that same organism in every single case of the disease. So you were able to find it in every case that had those particular signs and symptoms. In this case, the sheep has the lying down syndrome. Actually, anthrax is a very awful disease. Then you were able to isolate from a diseased animal microorganisms, and you found the same microorganisms from all of the diseased animals. So this is where Koch's lab really excelled. Koch's lab gave us many of the techniques that we still use today, that you will be using in lab, to grow bacteria in culture, to isolate them, to grow them in pure cultures, to do studies on them. So that's what they did, and that's what they spent a lot of time doing. And then postulate 3 is that you take this pure culture and then you expose a naive animal, one that has not been exposed to that disease before, to that organism, and then see what happens. So again, that animal gets sick with the disease, and then you re-isolate the same microbe that you put into the healthy mouse. You take from the sick and dying mouse the exact same microbe. So those are the postulates. And the problem is that in the real world, this works, you know, how well rules work. There are always exceptions. In fact, there are probably more exceptions than there are rules for this. But the point being, you know, here are just some of those things. So disease presentation for several diseases caused by microorganisms is highly variable, right? Some people get the flu really, really badly. Others get just barely a sniffle. Some people will die from tuberculosis. Others carry it around in their bodies for their entire lives. So it's sometimes very hard to verify postulate number one. Number two, they don't always lead to a disease that is disease, right? So you can be infected but not have the disease, like poliovirus, for example. Poliovirus is very common, but it only causes polio, which is a paralytic disease, in less than 1% of those people who are infected. So polio is scary, but only about 1% developed the paralysis that led to that could lead to the iron lung, right? Having to have assistance breathing, losing the use of your limbs. So there's that issue as well. Then number two, you have to, and 2 and 4 actually, you have to be able to grow the microorganism in culture, separate, away from all of the microbes and away from the body in an artificial environment. So we cannot culture most microbes. We can culture some of them. And in fact, the ones that can be cultured, we have cultured. We have grown them, we have studied them. But now we realize that that is a tiny proportion of all the microbes, and even the disease-causing ones, and even the ones that we know can cause disease can be very difficult to grow in culture. Viruses are an excellent example of that because you can't grow viruses in a culture, in a petri dish. You have to grow them in live animals. So that just complicates things. And then, lastly, if the microbe only infects humans, to infect purposefully somebody with a microbe to see if they will get the disease is profoundly unethical. Not that humans haven't done this in the past, but in the modern world, it is something that is forbidden and has been forbidden since the 1950s, roughly. So lots of ethical considerations to think of here. So the point being, it isn't always easy to identify a microbe as a cause of a disease. And I will tell you that there are many microbes that cause illnesses that are attributed to other things, chronic illnesses that are not attributed to a microbial cause, which are actually caused by microbes. So that's my stick on Koch's postulates. So throughout the golden age and then into the 1940s, into the 1950s, antibiotics became widely available. People stopped caring so much about bacteria. The focus in medicine turned to cancer, and then there's always viruses, which we still have very few effective therapeutic approaches for. But I just going back to that period of time, right around the end of the antibiotic, the beginning of the antibiotic era into the end of it, which was really like the 1980s, 1990s. And I want to give a shout out to these two guys, René Dubois, who is best known for, if you're a cell biologist and cell culture techniques, he did a lot with that. But he was the one who said, growing them in pure culture, growing bacteria in pure culture and doing these experiments with them in pure culture is not how they live in the real world. So it's like taking a tiger and taking it into your house and turning it into a house pet, or, domestic cats or dogs. They live domestically. And if you put a cat out in the real world, they wouldn't know how to hunt. They wouldn't know what to do. They wouldn't know how to survive. They would be attacked by other things and they wouldn't know what to do about it. That's really when you grow bacteria in culture, that's what you're doing. You're making them into pets, and you're nurturing them, and you're treating them kindly, and you're doing all these lovely things. And you put them out in the real world with other microbes who are not so nice and, to their own devices, and they don't survive long because of competition. So what he was saying is that we should take an ecosystem approach when it comes to studying microbes, and that includes diseases. So diseases were presumed to be caused by the microbe, but the reality is disease is complicated. So it's not just the germ, it's also what surrounds the germ, the overall condition, your overall health and wellness as well. So he wanted to say that, even humans are an ecosystem. It was not, he said it, but it wasn't fully embraced until later. And then I'll just give a shout out here to Joshua Lederberg as well, who said basically the same thing. He said, humans are not just humans. We are a composite of human and our microbial inhabitants, and they're not all bad, right? Obviously, they can't all be germs. Some must be doing good stuff. And so then there came the term microbiome, and Joshua Lederberg was the gentleman who gave us that term, microbiome. And so what he said was the microbiome is the ecological community of commensal, symbiotic, and pathogenic microbes that literally share our body space and have been all but ignored as determinants of health and disease. And welcome to the 21st century, because the 21st century is the era of the microbiome, and hopefully only the more good stuff to come out of that as we're finally moving away from the germ theory into the realm of the microbiome. Okay, so some of those relationships, ecological relationships, right? When one thing in ecology, when one thing lives with another, we call it a symbiosis. But there are many ways that we can live with other living things. So we can be mutualistic, meaning, the parties involved all get along and support each other and, do what they can to help and support each other. In the microbial world, microbes produce metabolites that other groups of microbes could use, which then produce other metabolites that other groups could use, that kind of thing. Commensalism is one where, one species benefits while the other one is not affected. And this applies to us because we are full of commensals. So, we're like a house for bacteria. They live in us, they kind of do what they want. They don't cause diseases. They don't necessarily contribute good things. They're just kind of there. And every once in a while, they pop up and it's just like, I'm going to do what I want to do, and they can cause some diseases. So, the mutualism is where everybody gets a fair share of the pot. Commensalism is where, perhaps the microbes are getting more out of that deal than we are. Parasitism is where one attacks and harms the other. Right, so parasitism and the word pathogen, which are harm-causing bacteria, applies to those. So these are the disease causers, right? The parasites are disease causers, but again, it's not black and white, because parasites, like I said, you can be infected with a parasite and have it live with you for your entire life without it ever causing you any harm other than to suck up your nutrients and resources. And then, the idea of antibiosis, this is where antibiotics come from. So out in the real world, bacteria are fighting, they're competing with other groups of bacteria because, there's only so much supply out there and they got to do what they got to do. So they produce antibiotics, they produce chemicals and probably other things that are able to inhibit the growth of their competitors. So this occurs in all ecosystems. And that, by the way, actually includes us as an ecosystem. So they come up with antibiotic methods or approaches or chemicals to, win the war, if you will, for themselves at the expense of others. So these are just some terms you should go back and review to make sure you understand all those relationships. And then realize, again, these are not black and white, these are not rules, because all of them break the rules. That's what life does. Biology breaks rules. So we try to, us humans like to try to say, well, this is this and that is that. And then the microbes come up and they just show you just how little we actually do know sometimes. Okay, so keeping along the line, the thoughts that of ecosystem, the human body is an ecosystem. Okay, so the human body is a wonderland, just to quote John Mayer. And in addition to that, so here we are, this is us. So here is us. We have cells, we have tissues, we have organs, which you have studied throughout your life as biologists and anatomy and physiologists, for example. So, but that isn't all of us. So you take us, and then you take this collection called the microbiota of bacteria and yeasts, and I'm going to include viruses in there, which you can see make up, there's tons and tons and tons of cells of microbes, in addition to our tons and tons and tons of cells of us. So you take this and you add that, and you end up with us, a complete human. So this is the thing that's kind of hard for some people to get. So in a cell, you know the concept of a genome. What's in your genes? Your genes are human DNA that's found inside of your cells. Well, guess what? Your genome, there's a metagenome involved, meaning all the genomes of all the microbes that inhabit you contribute to your genome as well. So it's your genome plus their genome that makes us. And that part is like, you know, wait, so bacteria are in control of my genes? And the answer is, yeah, and viruses, maybe even more so. And sometimes that, you know, it's sort of like, ew, I don't want bacteria in charge of my genes. But maybe by the time the course is over with, you will. So looking at, you know, where we've come over the centuries, right? So microbiology started in the mid-1800s with the isolation of bacteria in cultures, growing them and studying them as single entities, which obviously had limitations because only a percentage of microbes are in fact culturable. So that, and then in the 1980s, 1990s, we developed the ability to sequence DNA. So in other words, to remove DNA from any source, determine the sequence of nucleotides and determine what genes were present in that environment, maybe, and not even necessarily knowing what cells were present. You could just do that. And so now we can do that. We don't have to grow bacteria in culture to know that they are there. We can go fishing for their genes by fishing for this one little piece of 16S ribosomal RNA, the gene for it. Why do we choose that? Because it's specific for bacteria. Only bacteria have this gene because they're the only creatures that have 16S ribosomal RNA. So here's the eukaryotes, other organisms have and are a type of RNA also, but it's a different type. So we can use different probes to go looking for their genes. But now, instead of having to rely on bacteria showing up and growing in an artificial environment, we can actually just go hunting for their genes. And that's what happens very often. And that has led to our explosion in understanding in the microbes that exist in the world, especially when it comes to the human microbiome, right? So it was the sequencing of the human genome, which ended in 1999, that led to the realization that, wow, we don't have enough genes to be able to do everything that we do. And then realizing those genes are being contributed by the microbes, and that the technologies that were developed as a result of the Human Genome Project are now being applied to microbes. And that's a good thing for everyone. So these are some of the more modern methods, right? Gene amplicon sequencing, meaning just looking for that little piece, versus shotgun metagenomic sequencing, meaning take all the DNA and see what's in there, right? Looking at the whole thing instead of just going and picking out individual things, just saying, all right, what's in there? The only way these things work, by the way, is if you have computers with tons of computing power, and that artificial intelligence. And that's what had to happen before we could get to where we are now. So in the past decade. And then there are the omics, the famous omics. So genomics is the study of genes and genomes, right? Genetics. But you can also look at proteomics, even metaproteomics. So proteomics is looking at the products of gene expression, proteins. You can look at the transcriptomes, meaning the RNA, messenger RNA molecules that are being produced. You can look at the metabolomics. You can look at the metabolites that are being produced. These are usually done through mass spec or other highly technical chemical methods, which are done in research laboratories. So this isn't something that a little laboratory at a college such as ours is able to do, but we can do this. And there is still a place for culturing in the science of microbiology. And so, while this is what the techniques that we'll be doing in lab, realize that these other methods are actually out there and provide information, perhaps more information about certain things, but in the end, you can always go back to look at the culture methods. So I also wanted to just kind of highlight this idea that we humans kind of have parallel genomes. So we have our own genome, right? So we've got the latest estimate, like 25,000 genes in our DNA. So the DNA from our cells has about 25,000 genes, meaning we have the ability to express 25,000 different proteins or peptides. And then, everything that goes on from there. So that is our genome, roughly 23,000 individual genes spread out across, 46 chromosomes and, actually 23 chromosomes. And I can't even tell you how many billions of base pairs of nucleotides. So that's us. And, just when you look at what our genome is, all right, so, humans get our genome from both parents. So we have genes from one side on both sides. It is very stable. There is very little plasticity in it. And in fact, we don't want there to be plasticity. Mutations can cause diseases, right? There are several diseases in humans that are caused because of errors in the code that's within genes. So we don't really want our genome. We want it to be ultra stable, without flexibility, without alterations. Therefore, we have limited diversity in terms of our genetics. Gene modulation is very risky, right? So there's a lot of stuff going on right now in science where we're trying to change our genes as a treatment or for diseases, right? So let's change our genes. It will make us more powerful, more this, more that. But the changes are profoundly unpredictable because we can sit here and say, well, if we change that nucleotide, this should happen. And then again, at the end of the day, life does what it wants, which is a recurring theme for me. Biology does what it wants. Life finds a way. And I keep saying that over and over and over again. And if you don't want to know where that comes from, you should go watch Jurassic Park, because it says it very, very well. Okay, so then there's the microbiome. All right, so notice about the same number of cells. So we have a bunch of millions of cells. We have equal number of cells that are microbes. But notice those cells contribute 3 million genes to us. This is our microbiome, all right? So this is the genome of our microbes. And then we can put that with our genome, and that's called the metagenome. Okay, so we have two parallel genomes, but then they're not separate. They come together, and that's referred to as the metagenome. So, and the thing is, your metagenome is different from my metagenome, is different from everybody's metagenome. So there are similarities, but there are profound differences. And this is where diversity comes from. So, when you look at our genomes, every individual human has pretty much the same DNA, almost exactly the same DNA. So where do the variations come from? And it's now, becoming more and more clear that our microbiome is contributing to the diversity that we see between individuals even. So there is plasticity, there is variability, and can it be modulated by targeting interventions? I don't know, maybe not, but we don't at the moment. We have ideas of who the good guys are and who the bad guys are, and which ones we want and which ones take advantage of it, but we don't know the whole picture yet. So targeting interventions might be difficult, but it's something that is the, you know, the goal of the future is to research this and figure out more about it. Okay, so just to kind of sum it up, this particular article, if you want to go read it, it's open access. You could go read the whole article, but it just kind of discusses the microbiome in health and disease. And so basically, these are the major summary points that I've tried to make, right? Microbes, the big picture is microbes are an essential part of our biology, our life. They are critically important to health. There's this concept of homeostasis, which you hear about in anatomy and physiology, meaning that we have to keep everything in balance. Well, guess who's doing that? That's the microbiota, or microbiome is doing that. Okay, so we started off stable. They're the ones who are maintaining it. And so they're important in health. They are as important to our health as they are to disease. And we don't appreciate them. We do everything we can to decrease their numbers. We take antibiotics, which decimates their populations and causes all kinds of secondary problems, none of which was known in the 1950s, 60s, 70s, or 80s. And even now, even though we realize that bacteria have evolved resistance to antibiotics, we still, you know, looking for more antibiotics. We still don't appreciate that those antibiotics have caused damage have weakened us in some ways, just because they disrupt our microbiota to such a great extent. So that's something that has to be looked at in the future as well. And then you know, just the idea that it's, you know, where are these microbes? They're all over the place, but one of the key areas where all of this comes together is in our guts. So you know that your poop has bacteria in it. Everybody knows that. That's why we go to the bathroom in the toilet, because poop is associated with diseases, because there's bacteria that can be spread that way. Well, your poop is mostly bacteria, among other things. But it also turns out that the vast majority of those microbes are in constant communication with our central nervous system, and also through our endocrine system, through our central nervous system. So this is kind of what's referred to as the gut-brain axis, but it's the microbes in the gut and the immune system monitoring that occurs just outside the gut that is influenced by the microbiota in your gut. Notice over here, the release of neurotransmitters by bacteria that go up to your brain and give directions to your brain. These are bacteria giving signals to your brain as a two-way street. And then over here, a direct connection is through the vagus nerve. So note the arrow. I put that there for a reason. Okay, reason number one is that would be my favorite cranial nerve. And the reason why it's my favorite cranial nerve is because, number two, it is the way that your gut microbes, and probably other microbes too, send messages to your brain. And notice, again, it's a two-way street. So the vagus nerve goes all the way down to your gut, takes messages up to your brain, and sends messages back down to your gut. So this whole idea, it's the gut-brain, right, the neuroendocrine axis is all regulated by your microbiota. And when that gets out of whack, so notice the human microbiota in this idea that there is a healthy, you know, a good balance of microbes. When it's healthy, we are healthy. And when it is not balanced, then notice these are diseases that there are several published research studies on that show that the human microbiota in a state of dysbiosis, imbalance, can lead to all of these other conditions, right? Obesity, inflammatory bowel diseases, and actually others as well, liver diseases, infectious diseases. So interestingly, there was just a paper published that regarding Lyme disease, that the gut microbiota of patients with chronic forms of Lyme disease are different than the gut population of people who are healthy. So which came first? Did the gut microbiota lead to the chronic symptoms, or did that cause it? And that's the problem that we are having, because this isn't something that we can apply Koch's postulates to, right? So diabetes, atherosclerosis, right? So heart disease is related to dysbalances in your microbiota. Metabolic syndrome, that's type 2, pre-type 2 diabetics. So that brings me, to the idea that, the last idea here is that these two words are fairly new words in the litany of medicine and microbiology: eubiosis versus dysbiosis. So eubiosis means the microbes in whatever location, in the gut, because this is where most of this has been studied, are in the right balance. There's the correct number of gram-positive bacteria versus gram-negative bacteria versus these other types. And they're all communicating well, they're all doing things, they're all telling your brain the right stuff, and everything is working well, right? So somebody who has a eubiotic gut microbiome is going to be in homeostasis. And then happens dysbiosis. So how does dysbiosis happen? You take antibiotics, you are severely stressed out, you eat crappy food, because I hate to tell you, the crappy food, pizza, all this stuff, what we know that I know who feeds off that stuff. Those are the guys that like the quick hit, the fast growers. I mean, no offense to E. coli, but these are the E. colis of the world. They're the ones who are able to grow faster, grow, to larger numbers if you eat crappy food. And that crowds out the ones who are slower growing, maybe more methodical about stuff, more thoughtful, if you will, in the overall realm of that. So what happens is, right, these certain types of bacterial populations called firmicutes go up, and when they should not be, remember it's a balance. So when there's more of these firmicutes than there are other types, that tends to lead to this dysbiotic state, and that tends to lead to disease, right? Various disease states, which, they're finding out more and more that they're related to. So I'm not giving you a lesson on nutrition, but what I'm saying is that when you go here, you are promoting the growth of the fast growers, which promotes dysbiosis, which can lead to health issues later on down the line. That's the short version of the story, and it's still being parsed out, and that's where the research is going. So that's all for today, and next time we'll be delving into the secret lives of bacteria. Actually, not the secret lives of them at all, because we're going to be looking at those typical fast-growing bacteria that we know quite a bit about because we've been studying them for the past 100 years. So I will see you then.